We're continuing with photosynthesis now, and this is the depiction of what goes on in the first half of photosynthesis, which is referred to as the light reactions. And there seems to be quite a bit going on here. Don't be intimidated by it. We're going to break it down into simpler components and follow basically this series of events that occur. But at first glance, let's talk about where this is located and what we're looking at here. You recall from the last module, we were talking about chloroplast structure. Well, this is in the interior of a chloroplast, specifically this region above this line here where all these components are, which represents the thylakoid membrane. Remember, the thylakoids are the little stacks of discs, or the individual discs in a stack, which is called a grana, the interior of which is called the lumen, and then the space between these thylakoid stacks and the outer membranes of the chloroplast is referred to as the stroma. All right, well, light drives this process of photosynthesis. That's why it has photo in the name of it. And it all starts here in this region, which is labeled on this drawing as PS2, and that's short for photosystem 2. So following along in the notes, let's talk about photosystem 2, or PS2 for short. Well, photosystem 2, as it says in your notes, is a group of pigments, and we mentioned that in the last video as well. Photosystem 2 is very important because it gets the whole process going. But it's also extremely important to us as animals in that this is the part of photosynthesis where the oxygen gas gets formed. If you remember, our overall reaction was 6O2 and sugar as the products of photosynthesis. Well, here's where the first product gets made. Photosystem 2, again, is a complex of, or a group of pigments, which has lots of chlorophylls, also some xanthophylls and carotenoids. And in the middle of all of these group of pigments, there's one particular pigment known as P680. And P680, when it becomes energized by incoming light, becomes what's called a strong oxidant, which basically means that it can rip apart a water molecule. And if you follow the notes, it has the reaction of 2H2O being torn apart into producing oxygen gas plus 4 hydrogen with a plus sign means hydrogen ions, and an additional 4 electrons. And electrons we write as an E with a negative charge because they're negatively charged parts of atoms, and we can just leave it at that for now. So when P680 becomes excited, becomes energized, it basically chops up a water molecule, produces oxygen, and releases also electrons. And so if we look back up at our figure here, we'll see that's exactly what's going on. Here's our original water molecules. They're coming into photosystem 2. Light energizes these pigments, particularly that P680 stuff, which chops up the water molecules. You can see we can have an oxygen and 2 hydrogens together is water. Over here, it's been split apart. Hydrogens now are in the form of hydrogen ions, and oxygen atoms, once this happens a second time, will combine to form an O2 molecule. Now, what happens through the rest of this process, to figure that out or to understand that, we have to follow what happens with all of the electrons. Well, for each water molecule, we come back down here for a second, each water molecule has released from it a total of 2 electrons, and when we have 2 water molecules being split, we end up with these total of 4. So up here, we're following 1 water molecule. Right here, I've written it as 2 water molecules. So but the numbers aren't very important to us, rather than understanding what's going on in the overall process. So what happens next with these electrons? Well, the electrons, we can follow this arrow here. They get passed along a series of things here, and I just have them drawn as different shapes here. And these are various molecules that are part of what is called the electron transport system. And what is the electron transport system? Well, as outlined in your notes, it's basically a series of molecules, and their job is to pass electrons from one to another. And basically what happens is these electrons are full of energy. They're very energetic. And they're sort of like, if you recall, maybe when you were a kid, you played hot potato. Well, Photosystem 2 can't hang on to them or it gets burned, and that's exactly what happens if it hangs on to them for too long. And so instead of doing that, it hands them off to this guy, this red circle here. What the names of these aren't important to us for our purposes, just that these electrons get passed to this guy, who in turn passes it to this guy, this yellow one, who in turn passes it to this gray, blue, or whatever color that you want to call that is, and who's going to pass it to the next one. Well, this is called electron transport. So this flow of electrons occurs in this direction. And so electrons get transferred from photosystem 2 all the way over here to PS1, which, as you might guess, is called photosystem 1. So electron transport molecules will say move or pass along electrons. Remember, E with a negative is electrons, from photosystem 2 to photosystem 1. Now, one step along the way, as you might have noticed here, does something beyond just play hot potato with electrons. And this guy right here takes a little bit of the energy that's inherent in those electrons and it uses it to do something. And what it uses it to do is it uses it to move some hydrogen ions from the chloroplast stroma on this side of the membrane in and through the membrane, which they normally cannot pass through the membrane. Remember, membranes are semi-permeable. This not being water can't just pass through the membrane, so it has to be helped across. And so basically, this guy in the middle here takes some of the power from these electrons and uses it to pump hydrogen ions from this side of the membrane to this side of the membrane. And so, as you can see or maybe visualize now, we're starting to build up quite a few hydrogen ions from not only the splitting of water, but also this pumping action of the electron transport system, to where we have higher concentration of hydrogen ions on this side of the membrane than we do on this side of the membrane. And we'll come back to that eventually in this whole process. So, electron transport system passes electrons. It also pumps some hydrogen ions into the lumen. And then finally, I'll say about electron transport, although we're not naming them, it's important to note that there are several of the essential nutrients that are essential components of these electron transport molecules. For example, many of them contain sulfur, iron, and also, I believe, manganese. And so this is where some of these essential nutrient roles come into play in this electron transport process. Now, finally, about electron transport, let me say this in regards to the hot potato statement. If one of these components breaks down or is not in the proper place so that it can pass these hot electrons along and get rid of them, they're left stuck. One of these is left stuck holding electrons, and these highly energetic electrons will burn basically one of these complexes up. And when that happens, you start to get damage going on inside of the chloroplast, which is not a good thing for the plant, because if the chloroplast is damaged, it can't carry out this process, as we'll see. The membrane itself being damaged by these electrons, if they are stalled in this process, prevents the buildup of these hydrogen ions, which we're about to see is quite important to this process. So that's electron transport. Next in line is photosystem 1. Photosystem 1, like photosystem 2, is just a group of pigments, in the heart of which is one very special pigment called P700. P680 was the special pigment in photosystem 2. Now, the name P700 just refers to the fact that its peak absorption is at a wavelength of 700 nanometers. Just like the P680 in photosystem 2, it has a peak absorption of 680 nanometers. That's all the numbers refer to. Instead of splitting water, P700 basically adds more energy to the electrons that it receives from the electron transport system. So it becomes excited when light hits, as we have it depicted here, coming into photosystem 1. And it transfers that energy, and I'll go ahead and write that out, transfers energy to the electrons. It then passes the electrons, put a comma, to the next electron acceptor in line here. And this one I'm going to actually name, and it's called ferredoxin. Ferredoxin contains, as you might guess, Fe, which is symbol for iron, and it does indeed contain lots of iron. So there's an essential role specifically for iron. It's right here. In fact, I'm going to go ahead and write that in. There is iron in this guy right here. Okay, and that's called ferredoxin. So, following along, the electrons started in water. They got split by photosystem 2. They got passed along, shuttled some hydrogen ions along the way. Photosystem 1 takes them and gives them a little last boost of energy and passes them off to ferredoxin. Ferredoxin then passes it to the next guy in line, which is called NADP reductase. So let's talk in detail about that guy. NADP reductase is an enzyme. And an enzyme is basically a little molecular machine, is a good way to describe it. And enzymes get named based on what they do. And this one is called NADP reductase because its job is to reduce a molecule called NADP. And in chemistry, when something is reduced, that means that electrons were added to it. And so basically that's what it does. NADP reductase, we'll say, takes electrons from ferredoxin and adds them to a molecule called NADP. Now this NADP stuff plus an electron plus hydrogen forms a molecule called NADPH. So we'll say 2 NADPH and hydrogen ion to form NADPH. I'll draw a little square around that guy. All right, that's all it does. And it's important to note, however, that this NADPH is then released into the stroma. We'll come back to him later on. But that's it for NADP reductase. The last guy in line here is this guy. And this guy is labeled as an ATP synthase. So let's talk about what he does. An ATP synthase is another enzyme complex. And like the one we just talked about, it's named based on what it does. And what it does is it synthesizes or makes a molecule called ATP. We've talked about ATP briefly before. This is referred to as photophosphorylation because of the way ATP is made. This combines, as it says in your notes, ADP with phosphorus, and that's what this little P in the yellow globe here is. That's PI for short, called inorganic phosphate, basically is what that means. And this forms this molecule called ATP. So basically ADP, which is short for a molecule, you don't have to know this, called adenosine diose phosphate, plus an additional phosphate becomes adenosine triose, meaning 3 phosphate. So diose phosphate plus another phosphate becomes triose phosphate, or ATP. And I always draw a little comic book explosion around ATP because ATP is an energy carrying molecule. And so ATP synthase basically makes ATP, which contains quite a bit of energy. Well, where does the energy come from? Well, that's where what we've been building all along, this hydrogen accumulation in the lumen comes into play. If you remember back when we talked about diffusion and osmosis and how things like to move, well, we've created, from square brackets mean concentration, right? Hydrogen ion concentration on this side of this membrane, which is relatively high, and let me get enough room up here, a hydrogen ion concentration on this side of the membrane, which is relatively low. And so hydrogen ions on their own naturally want to move from regions of high to low. But why can't they? Well, because this membrane, although it's not drawn all the way to the edge of the sheet here, this membrane continues and is continuous. And so they are separated from this region of low concentration. However, an ATP synthase is shaped just so that hydrogen ions can fit exactly through the middle of the enzyme. And that's exactly what they do. As long as there's a gradient from high to low, the hydrogen ions will naturally want to flow. And so you can think of this as sort of being a little tap into the chloroplast lumen. And although it's not truly pressure, the gradient kind of acts in the way of pressure, in that it basically allows them, or makes them want to anyway, move to the other side of the membrane. And that's exactly what they do. And an ATP synthase is set up in such a way that it can harness the energy from that flow. It would be sort of like, say, a pressurized bottle, or some sort of a rocket engine. Well, pressurized gas escaping through the rocket engine provides thrust. Well, in this case, it's not providing thrust. It's taking that energy, and instead of moving something around, it's putting it into the reaction of the formation of ATP. So, to summarize all of that, hopefully you're still with me. ATP synthase, it makes ATP in this reaction, and it basically runs on the flow of hydrogen ions from the lumen, where they're high concentration, to the stroma, where the concentration is low. Let's summarize, and you can follow this in the notes. Hopefully, with a little bit of studying and becoming a little more familiar with this module, you'll be able to look at this and basically explain what's going on. So what is going on? Well, we're looking at a thylakoid inside of a chloroplast. We've got our thylakoid membrane here, which separates the lumen from the stroma. Light comes in, photosystem 2 absorbs that light, which allows it to chop up water molecules, producing oxygen, which is important for us. I'm going to circle this as one of the products of the light reactions. And it takes these electrons and passes them through the electron transport chain, or electron transport molecules. Next stop is photosystem 1, which boosts the energy of the electrons further, passing it to ferredoxin and this NADP reductase. NADP reductase adds electrons to NADP and hydrogen ions to form NADPH, which is another product of the light reactions. This ends up in the stroma. The remaining hydrogen gradient is then put to use, or simultaneously actually put to use, to run an ATP synthase. And the ATP synthase catalyzes this reaction, ADP plus P, making adenosine triose, or ATP triose phosphate, which is the third product of the light reactions. Now, this is only half of photosynthesis. This is the most complicated half, so you can, if you are still with me, breathe a sigh of relief. This is about the most complicated thing we'll cover in this entire course. But bear with me, we still got to talk about what happens through the rest of photosynthesis, and that is referred to as the dark reactions, which take place out here in the stroma, and they are basically fueled by NADPH and ATP that we just produced in the light reactions.